our final chapter, genetics and genomics, which is a related field. So, some definitions. There's a lot of vocabulary in this chapter, but hopefully the concepts are interesting enough that it'll balance that out. So, genetics is the study of inheritance, how traits are passed on. Genes are sections of DNA that code for a protein. And the genome is all the DNA for a species, but it's one set of it. So the human genome is 23 chromosomes. So with that little extra X or Y, there's 24 different possible chromosomes. So genomics does the wider studies of what's going on with the genes beyond just how something is inherited from one generation to the next. So a gene would be one portion of a chromosome made up of many base pairs. DNA is a language that can be translated into protein. It's like Morse code. You might remember some of this from back in chapter 4, where you have a small number of terms, A, C, G, and T, that combine, and those combinations give you more information, which in this case is 20 different amino acids. Unlike Morse code, where it can be any length because you have a pause to designate the end of one letter, the genetic code is always 3 bases for one amino acid. And then the sequence of amino acids determines what protein you get. Somatic cells is the term for body cells not including eggs and sperm. They are what's called diploid, which means they contain 2 copies of the genome. We call gametes haploid for having half the DNA. So there's no term for having one set of DNA. We call that half because it's half of what the somatic cells have, but we call the somatic cells diploid for 2 sets. Your somatic cells got one set from the egg and one from the sperm that made you. And so we all have 46 chromosomes, barring any variations that we'll talk about later in the chapter. In the human genome, there's a little over 20,000 genes. Each cell only uses some of those genes. But each gene can code for more than one protein because after it's copied into messenger RNA, that can get edited, and then different pieces being cut out provides different proteins. So there's somewhere between 100,000 and 200,000 different proteins from these 20,000 genes. Gene expression, another reference back to chapter 4. This is building the protein. It is regulated by neighboring parts of the DNA. So if you have this strip and here's the gene, some sections before and after, especially before, would control when that gene gets transcribed, copied into messenger RNA. Factors that affect the transcription include hormone levels. If you recall, thyroid and steroid hormones often alter gene transcription. Environmental influences, is this protein needed right now? And interaction with other genes. Some genes can say, well, we're making this, so we also need to make that, or we don't need to make that. Questions so far? modes of inheritance. This is one of the fun parts. This is where you get to actually look at each other and yourselves and your family and think about real examples. Modes of inheritance are the ways traits are passed. There's a basic pattern that was discovered by Gregor Mendel, a monk, in the 1860s. He was working with pea plants. They are also diploid, so the same principles apply to us and to anything else that's diploid, that has two sets of chromosomes, one from each parent. Of our 23 chromosomes, 22 are called autosomes. That means self-body. These are the same regardless of sex. And then the last pair is the sex chromosomes, X and Y. We'll talk more about these coming up. Y actually has a gene on it that causes an embryo to develop as a male. And that's why you have to have a Y to be a male. It's not just, here's this chromosome. It's, this chromosome is here, and this gene activates. A karyotype is a picture of the chromosomes. They line up the autosomes by size, and that's how they're numbered. So, number 1 is the largest, although I think there was some error early on that we stuck with, and the 21 is actually a little bit smaller than 22. And you'll see why that matters when we get into genetic disorders. And then the sex chromosomes are listed last. Can you think of anything that we can tell from this?
Sex of baby.
Yeah, you can tell the sex. What's this one?
Male. Female.
Yeah, this is female because those are the same. Female is 2 X's. Okay. Have you heard of chromosome disorders? Say, yeah, Down syndrome, that's going to relate to chromosome 21. That has an extra. So if you could do a karyotype, you would know for sure if somebody had Down syndrome. There are some other cases where there can be extra chromosomes or missing chromosomes or pieces that are added or deleted that can cause different disorders. Oops. There. Those pairs are the homologous pairs or homologous chromosomes. They have all the same genes in the same places. Say something like that, although there would be a lot more than just 5 genes on each chromosome. Each different form of the gene is called an allele. So a different form of the gene would give you a different form of the protein. If both copies of the gene are the same, then it's called homozygous, same pair. If you have 2 different alleles, that's called heterozygous, different pair. The genotype is which alleles you have. And that would be shown as 2 letters, or sometimes it's multiple letters or another kind of symbol. The phenotype is what trait you have, and that comes from your genotype. Some terms that are used here, wild type means the most common or the natural form. This is either the most common phenotype or it's a protein that's functional. You hear about this a lot if you do fruit fly genetics. Like the wild type fruit fly has red eyes and a yellow and black striped abdomen. And there are some variations or mutants that might have white eyes or black eyes, or their abdomen is all black. So a mutant is any change from the wild type. Some mutations can cause disease. And that would be the case where the wild type protein is functional, the mutant one isn't. And so the cell it's in loses the function of that protein. Okay, so far? Dominant and recessive are major inheritance patterns. Dominant means that you only need to have one copy of the allele to get the trait it codes for. So if you're heterozygous, you'll have the dominant phenotype. We indicate dominant alleles usually with a capital letter. So something like, let's say, dimples. Having dimples is dominant, so it's a capital D. Not having them is recessive, so we use a lowercase d. We always use the same letter for a gene. If we have to be more specific than this, like if there's more than 2 variations, we might add a subscript or superscript to specify, or sometimes there'll be multiple letters. Recessive, the phenotype only appears if you are homozygous for it. So since you have 2, then your possible genotypes are homozygous dominant, heterozygous, and homozygous recessive. So only this one would have the recessive phenotype, because you only need one copy of the dominant allele to get that phenotype. Questions? So in order to have a dominant phenotype, one of your parents must have had the dominant phenotype, because if they had the dominant allele to give it to you, then they were expressing that trait. Sometimes a new mutation can be dominant. It's not very common, but it can happen. For recessive alleles, the phenotype can appear when neither parent has it, because they could both be heterozygous. And then they happen to both give the recessive allele to the child. And so they get the recessive phenotype. Heterozygous is sometimes called a carrier. This is especially for diseases where you're not sick because you have the functional protein in the form of the dominant allele, but you could pass the non-functional one on to your children. So, when we talk about something skipping a generation, it's usually something that's recessive, but it's not always that predictable. It's not like grandparent and grandchild have it, and then that one's grandchildren have it, and the ones in between don't. It can turn up in any generation as long as the allele is still there on both sides. Dominant and recessive making sense? Okay. To add to that, where the gene is affects the pattern of inheritance. An autosomal gene is anywhere on chromosomes 1 through 22, and that means you get all of that from both parents, and so both sexes are affected equally by the trait, or is found equally in both sexes. An X-linked gene or X-linked trait is found on the X chromosome. For a recessive phenotype, that's more likely to show up in males because they only get one copy of the gene. They get an X and a Y, and most of the genes on the X don't have a match on the Y. So you have, if you get one copy of the recessive allele and you're male, well, for anything, if you get one copy of a recessive allele and you don't have anything else to go with it, then you have the recessive phenotype. Where for females with 2 X's, you would have to get the recessive form from both parents in order to show the traits. So X-linked is more common in males. Y-linked the gene is actually on the Y chromosome and has no mate on the X, these are traits that can only occur in males. Or at least they can only occur in somebody who has a Y chromosome. There are some ways that you can have a Y and still be female. So a Y-linked trait is normally only passed from father to son, where an X-linked trait, a son would get it from his mother, who either has the phenotype or is a carrier. Questions on sex-linked traits? So here's some examples. Now we're going to go into the eyes example, and it's actually more complicated than this, but brown is dominant. Basically, the melanin in any location is dominant. So you will often see here, there's brown-haired parents and one child